All right, everyone. Well, welcome to the first Family and Researcher Roundtable. My name is Casey Craig. Many of you know me as Stella's mom, but for those of you that are new to our community, I am the Executive Director for the Acute Syndrome Foundation. As you remember, over the last 3 years, TCSF has hosted a fall education series, but we called it Clinical Trial Readiness. But we kind of realized over the last year or so, it's really time to pivot that focus now that we have 2 trials currently going on. And so we're going to host a variety of research and quality of life topics to keep our community informed and active in the care and treatment of SCN8A. So before I introduce our amazing guest, I want to kind of go over a few housekeeping. So this is a 3-part series. So clear your calendar for the next 2 Wednesdays after this. I know we're Thursday today, but we'll be 2 Wednesdays from now from 12 to 1 p.m. Eastern Standard Time. We will have these recorded so we can go back and listen to them again. I know, especially with some of the research heavy content, it's nice to sit and ask questions and maybe come back to it and listen to it again. We really want this to be super relaxed and comfortable, so feel free to ask questions. The chat box will be ready to go, and we have a very well-rounded Zoom user here with Madeline to be able to kind of keep track of the chat box and stuff like that, so this can be interactive. Due to screen size and background noise, if you guys want to keep yourself muted and your cameras off, that way we can have as much space for the presentation today. I also wanted to thank our platinum sponsor, Neurocrine Biosciences, for sponsoring this series. Okay, so without further ado, let me introduce our rock star SCN8A mama and researcher, Dr. Madeline Oden. So Madeline, I am so excited to have you on today because not only are you just this wealth of knowledge, but you also walk in our shoes every day with your daughter, Margot. And so do you want to tell us a little bit about yourself and then jump into today's topic?
Yes, yeah. Thanks for having me. It's great to be here. And I welcome any opportunity to interact with the community and share about the science and help break things down to make things more accessible for parents, because I think it's really important for parents to be knowledgeable about all that's going on, even though I know it's a lot to learn about and to understand. So I'm mom to Margot, who's 2.5, who has 2 mutations in SCN8A and has pretty severe developmental delays, low tone, and all that comes with that, daily seizures. It's on a bunch of drugs, and but is a happy girl who loves to swim in the water and go outside. And so my job, I'm a researcher. I'm an associate professor at Tufts, and my research background, I actually did my PhD in neuroscience, working on how cells move around in the brain, and then moved to cancer research for my postdoctoral work, and then started my own research lab at Tufts in 2018. That was initially only focused on cancer, although looking at some of the similarities between cancer and cells of the brain. And when Margot was diagnosed, I knew that I had to, jump in and do something about it. I couldn't just sit there and do nothing, because while I couldn't control her seizures or anything, I could do something in the lab, and that really helped me cope and feel like I can do something. And so with my husband, who's also an academic, and an awesome collaborator at Tufts. It turns out Tufts is very strong in epilepsy research in their neuroscience department, and have several labs that work on epilepsy. And one of my graduate students was dating someone who was in an epilepsy lab, and so I was able to connect and kind of work and get started in this community. And everyone's been really welcoming and helping us kind of learn more about the field. And so now we've been doing research, for 1.5 years in this area, and specifically focusing on ASOs. And so that's really what I'm going to talk about today, kind of trying to understand what ASOs do, how do they fit into kind of all the different treatment approaches that are out there, and what could work for SCN8A, because it's a unique gene, and so not every approach is going to work. Mm-hmm. So please ask questions along the way. You can put them in the chat, and I'll try to pick them up along the way. Happy to talk more about this too, and I'll be talking a bit about this too at the gathering in a month now, not even, so really soon. And so, happy to talk more about it then as well. All right, let's get started. Okay, so to kind of put things into context, back to some very basic biology of what we call the central dogma, which is what, the properties within cells that allow our cells to function. And so the DNA is the basis of any cells, and we all have DNA in our cells that is unique. And the genes are in the DNA, and that is where SCN8A, the gene, is located in the DNA. But DNA itself doesn't actually do anything functionally in terms of making the cell work. DNA gets turned into this intermediate called mRNA, and then mRNA gets turned into protein. And proteins are the effectors of cells. They are what get things done. And the protein name for SCN8A is NAV1.6. So they're different names, which is confusing. Some genes have the same name at the protein level, some don't. Of course, with SCN8A, it's confusing too because it's 8 and 6, but that's just how it is. So, and so the SCN8A gene is located on our chromosome number 12, and we each have 2 copies of each chromosome. One that we inherit from the mother and one from the father. And so we have 2 copies of SCN8A gene, and both of these make the protein. And so when mutations happen, they happen in the DNA, and that then impacts the mRNA and impacts the function of the protein. So what do the mutations have effects? And as you know, now, it's really important to understand kind of the effect of the mutation on the protein function, because different mutations can have different effects. And it's something that, we're starting to learn. And there's been a recent paper from the Hammer Lab in trying to predict based on the mutation, which one is gain in function or which one is loss of function. And, that is something that's important to understand. And so for some mutations we know, some we don't know, some we can predict. So gain of function mutations generally mean that one of the copies is mutated. It's always just one. And they're both functional, but one is much more active than the other. So the mutated one is more active, gain of function, so it increases the function. And so there, therapeutically for these patients, what we're trying to do is reduce the activity or the amount of SCN8A, right? To bring it back to normal because there's too much. On the contrary, for loss of function mutations, there what happens is there's a mutation that affects the DNA that leads to less protein or no protein being made from this copy. So you only have one functional copy. And so there, the therapeutic goal is to increase activity or the amount of SCN8A. So, generally gain of function is more associated with seizures, more severity in terms of delays and functions. And loss of function tends to be more associated with the more autistic or behavioral, but still can cause seizures. So it's not so black and white, and we're still learning a lot about these. And unfortunately, some mutations can cause both gain and loss of function effects on the protein. So it's not always that straightforward. But generally, this is kind of how we characterize mutations now, and this can still help kind of to figure out what the best approach is in terms of a therapy. And so we're looking at different things depending whether you have a gain or whether you have a loss. So what are kind of therapeutic options for, this is not, for any kind of disease that is caused by mutation in the DNA. So at the protein level, the way that we target the protein is by drugs or small molecules, right? These are our sodium channel blockers. And so obviously we know that many of our children are on sodium channel blockers that are not specific, right? For NAV1.6, for SCN8A, they affect lots of them, but having a specific one is, what Praxis and Neurocrine are doing. And so that, might inhibit the activity of the protein at this level. It doesn't touch, anything that's happening earlier on in this process. Actually, before I go, there's a question, are most SCN8A mutations gain of function? I think right now, the current estimate is about 20 to 30% are loss of function. But I think because loss of function is not always associated with seizures, there has not been as much of a push in individuals that might have a phenotype associated with loss of function to do sequencing. And now, with like the Simons Foundation, for example, there's a lot of push to sequence individuals who are autistic or who might have, intellectual disability that is, not related to seizures because there's likely a genetic cause. So I do think that the population of loss of function individuals will increase as kind of more sequencing becomes available and is encouraged for that population. So I think the bias has been towards gain of function because it's people with, seizures who tend to get sequencing. So at the DNA level, there are ways to intervene. And this is generally what is referred to as gene therapy. And gene therapy can mean a few different things, but generally this is therapy aimed at the gene, at the DNA. And so this would be potentially adding, replacing, correcting the mutated DNA so that you could then make a corrected protein. And then ASOs, which are antisense oligonucleotides, and kind of a newer technology that was first approved in patients with spinal muscular atrophy in 2016, is looking at mRNA. So the ASOs target mRNA, and they can be used to degrade the mRNA, to change it. And if you change the mRNA, then you change the protein. So those are kind of the three approaches that, can be used to, target, any rare disease, but in this context of SCN8A, any disease that is caused by a mutation. So again, at the protein level, we do have these sodium channel blockers. I'll talk a bit about these two interventions. Now, it's important to keep in mind in terms of these approaches, how easy they are to do, how easy it is to actually get these to the brain, because the brain is a well-protected organ in our body that has this blood-brain barrier around it that prevents a lot of things from coming into the brain, which is a good thing for us, but that can make it really challenging to deliver things to the brain. So in terms of kind of how these therapies work, so if we look at drugs, right, we know most of our children are on drugs that target the protein. You usually can take these orally, and it's pretty easy at this point to get drugs to get to the brain. Chemists have done a lot of work on this, and so getting drugs that can hit the brain is doable. The only thing is that they only last a couple of hours in the brain, right? That's why our kids have to take drugs multiple times a day. They don't last very long. On the other end of the spectrum, if we were to have a therapy that would target the DNA, this is something that would be delivered in the bloodstream by IV. And this is really hard to get to the brain. It's really hard to get big pieces of DNA to the brain and get them working. So this is something that is really, really, really challenging, especially for big proteins like sodium channels like NaV1.6. So this is really hard to do. But if you can get it there, it would be like a one-time shot, get new DNA in there, and that should get into the cells and be present in there. So it'd stay a long time. ASOs, on the other hand, are kind of in the middle. And so I gave, an example here of a gene therapy. This is Zolgensma. This is, again, for the spinal muscular atrophy. And this was one of the first ones approved for a disorder that can affect the nervous system. And there are a couple of rare diseases where people are starting to do some injections even directly in the brain. This is still very, very early and can be very, very challenging to do. The ASOs are kind of in the middle. So ASOs are attractive because you can inject them into the spinal cord, and from the spinal cord, they can go straight to the brain. So this, is relatively easy to do under anesthesia, in a clinical setting, but, is still straightforward in terms of getting to the brain. So it's, pretty easy to get there. And this lasts about a few months. So it's any treatment with ASOs, which again, Spinraza was the first ASO treatment that was approved for spinal muscular atrophy, and patients need to get injections every few months with these. So again, we're dealing with these different challenges. So I see a question of who does these therapies. So, I mean, for SCN8A right now, we only have these drugs that are currently being developed in clinical trial. Gene therapy is, I'll get to that in a second, is not something that anyone is actively working on at all. And ASOs are starting to be worked on, and I'll now highlight kind of where things are at later on throughout the talk for SCN8A specifically. So, so then what treatments work for who? And so I see a question too in terms of, how do you know if someone is gain or loss of function? So some mutations that are where there are multiple patients have been characterized in scientific publications in using methods in a lab to understand, does this mutation increase the function or lower the function of the protein? And so there are some ways to kind of look that up if there are other people with the same mutation. Al George, who's a researcher, is also coming out with a paper pretty soon where he's characterized quite a few more mutations as well. And so, but again, if we don't know, then you would have, as you say, a variant of unknown significance. So, so that's where Dr. Hammer has come up with a computational predictive model based on a range of parameters where they can put in your variant and different features of when you started developing seizures, different kind of aspects of how the child might present. And they have a tool to be able to predict whether this is likely gain or loss of function. So there is a publication on that. Perfect. Yeah. And both will be at the gathering. So those will be great and good ways to find that out. Again, mostly this is not like a hard stop rule, but if your child does not have seizures and is more presenting with behavioral delays and autism-like features, it is likely much more a loss of function phenotype. If your child is really severely delayed and impacted with lots of seizures, it is likely more gain of function. When it's in the middle, it can be harder to know, and it's not hard and fast. We're still learning a lot about these different features. But so for a gain of function mutation, when you have increase in function, again, you have more active protein that is needed. Your therapeutic goal is to reduce the activity. And so potentially all these three options could work. We know drugs can work to try and reduce the activity. ASOs can be applied to this, and potentially gene therapy to correct the mutation here in the DNA. This is using a new technology called CRISPR. That can be used to precisely edit DNA. This is still far, far away from being used in patients by any ways. Miriam Meisler's lab at the University of Michigan is doing some really nice experiments in mice to look at this and to understand what is possible in terms of this technology in mice. But translating this, is really quite a ways away. But for this approach, you can't just put in the corrected gene as like a new gene you're bringing in, because you already have enough of this protein. You don't want to give more of it. So the only way gene therapy could work for gain of function patients is mostly by correcting, which again is quite challenging. On the other hand, for loss of function, where you only have one copy and your goal is to try to increase, here a gene therapy where you're bringing in more SCN8A DNA to make more protein to bring it back to normal, that could be very attractive. The issue with this is that SCN8A is a very big gene, and getting big genes into the brain and functionally in cells is really challenging. There is some ongoing work in the SCN1A space in Dravet, which is mostly a loss of function disease, and some work, but still mostly in mouse models at this moment, showing that there is a lot of work ongoing to try and get big genes like this to be brought into the brain. So this is, ongoing work really at the research level, not really something right now that, could be easy to translate, and no really ongoing trials or any kind of thing in terms of sodium channels. But ASOs are also very applicable to this, and I'll give more specific examples throughout the presentation to try and get more activity. Right now, all the drugs that we have mostly decrease the activity of NaV 1.6, right? Reduce it back to normal. So there are really no drugs specifically for NaV 1.6 that can increase its activity and to kind of work at that level. Again, patients with loss of function are on many other drugs to try and regulate the overall activity in the brain, but that is not specifically through NaV 1.6 and targeting specifically this population. All right. So different treatments depending on the type of mutation, but ASOs are amenable to both, and that's because ASOs can do lots of different cool things. So how can we use ASOs to regulate the SCN8A, how much of the protein is actually made from the gene? So again, ASOs work at the mRNA level. So they can work by reducing the protein levels by degrading mRNA. So this would be bringing down, so reducing the levels of NaV 1.6. So this would be good for gain of function mutations. But it can also work by increasing protein levels, by increasing the mRNA first and then the protein. And so this means that ASOs could also work for loss of function mutations, different ones, but they could work as well. And lastly, they can change the protein by changing the mRNA. And this can work for mutations in specific regions of the gene. And so I'm going to go over these different examples, the pros and cons, where things are at, and what could be done. Okay, so the first is using ASOs for their ability to degrade mRNA to decrease the protein that is made. Again, this is for gain of function mutations. So the first thing that was developed was an ASO that targets the SCN8A mRNA. It is not mutation specific. So this targets both copies of SCN8A, not just the mutated one. And the goal and what they showed was that this could decrease the NaV 1.6 protein. So bring it back down to a more normal level to see effects that way. So this is work that was done by Miriam Meisler's lab in collaboration with Ionis Pharmaceuticals, which is the company that developed the Spinraza ASO that was approved for SMA, and that is really the leading experts in ASO technology. And they showed in mouse models that if you treat with an ASO, so these are survival curves for the mice, and so every time the line goes down is the mouse dying. And so in this mouse model of SCN8A, the mice die without any treatment, die mostly by 2.5 weeks. But if they die of a seizure and status, if you give the ASO and you give more, it extends their life, quite a bit. So this is obviously really exciting. If it reduces the non-mutated DNA, is the benefit reduced? I will get to that in a second. Yes. So because it's not specific, I will highlight that this is a similar approach to what is happening in SCN8A, right? You may have heard that Praxis is doing a clinical trial for an ASO for SCN8A. This was also developed. This was developed by Steve Petru's lab, also in collaboration with Ionis, where they created an ASO that reduces the levels of SCN8A. Again, you can see in this mouse model of SCN8A extends the lifespan of these mice quite a bit. It is not specific to any mutation, so it works for any SCN8A gene. And this is ongoing, and they've released some of their first data, and they already see seizure reduction within within the first injections. This is really exciting. But why are they not doing this for SCN8A, right? We know Praxis cares about SCN8A. And the problem is that SCN8A is a much more complicated gene than SCN8A. SCN8A is a much more important gene in the brain, and so the brain cannot afford to lose a lot of it. It's what's called a Goldilocks gene, which if you have too much or too little of it, it's unhealthy for the brain. So if you have two copies that are wild type, as in healthy, you're good, right? That's what we want for your brain. If you actually remove, both copies of SCN8A completely, the mice die really young, not possible. You can't live without SCN8A at all. You need to have some SCN8A to be able to live. We know that a loss of function phenotype, as I mentioned, where you lose one copy, there's lots of presentations with intellectual disability, delays, autism, hypotonia, and it can cause seizures too, right? This is our loss of function phenotype, which we're learning a lot more about. But this can cause lots of other symptoms and issues within patients. If you have one mutation, again, you can have seizures, and again, really nice work from Miriam Meisler's lab. They took a mouse and they turned on the mutation at different levels, and to see how much do you need of the mutation of the SCN8A to get seizures. And they found that for these mutations they looked at, only if you have 10% of SCN8A, you can have of mutant SCN8A, of gain of function SCN8A, you can have seizures in a mouse. So there's a very, if you have little too much or little too little, there's effects that can happen. And lastly, we know if you have a full gain of function, you have lots of seizures and stuff. And so the thing with SCN8A is that the window, this green window, which is the therapeutic window, which is what flexibility can the brain handle of different levels of SCN8A and different activity, is tiny, right? It's very small. And for SCN2A, this window is much bigger. So it's a lot easier to play around with the levels in patients, because if you move around a little bit, nothing will happen for quite a while until you get to these more, kind of more extreme levels. Versus with SCN8A, only if you change a tiny bit, there can be lots of effects. And so, unfortunately, this makes it really scary for any company to take this on as a project, because being able to develop an ASO is, really dependent on making one that really only turns things down like a tiny bit. Could this window be patient specific? Yeah, great question. It could be, for sure. It could likely be mutation specific. And so, we don't know. We have these experiments haven't been done to this level with a bunch of different mutations, so it could be. But generally, this is just something that clinically is like really hard, and lots of companies don't want to do. The other thing is they're also worried about the FDA, like not really wanting to accept a treatment that could take, a patient from here to here potentially. And, that's just not like a good outcome, and that's too risky for the FDA to approve. So, having spoken extensively with Ionis and Praxis, they are not interested in doing this. It's not that maybe other companies might not, but this will be really challenging to develop as a treatment for a broad population for our children.
I just want to jump in, and I'm really glad that you brought this up, and it's a very clear diagram that kind of gives some context to this, because I know it is frustrating when we're seeing other sodium channel mutations that we a lot of times will associate ourselves with. You're like, okay, well, if Praxis can make a drug for both of us, why can't they make an ASO for both of us? And so I'm really glad that you pointed this out. And I think it just gives, all of us families some context of why aren't there ASOs available for 8A at this point in time? And it's not that no one cares, but it seems to be very, very tricky, obviously.
Exactly. So don't worry, I have more options, so do not despair. So yeah, Taylor, what about targets of modifier genes? Yeah, absolutely. I mean, for sure, that's an option for sure, but I don't think we know enough about the biology to be able to develop that on a larger scale that could be applied for multiple mutations. So the second approach I want to talk about and is developing allele specific, so specific for a mutation. So you can develop an ASO that would then be for, So obviously this would mean developing a bunch of treatments for all the different mutations that we have. This is not easy to do. Each ASO for a different mutation would be a whole, different clinical product. This is what the NLORM Foundation is doing. You can apply to be accepted, and they will develop an ASO for your specific mutation to tune it back down. But here, and we applied to this foundation, and Margot is in there, but they would not do this approach. Okay, so for SCN8A, so even for a specific mutation, again, because even if you remove all of this and you get down to a loss of function, then you're turning a gain of function to a loss of function. And it's, even though, there could be, and there have been discussions about, whether that is better and whether a family would actually want that, if you have a severe gain of function kid, to turn them into a loss of function child that may have not as many seizures and more development. FDA wise, regulatory wise, that's just not something that is feasible. So, that again, and Lauren, would not accept Margot or other SCN8A patients for this approach. So no one is really willing to go there at this point in any way in terms of developing a specific ASO for this mutation in the gene. So that's ongoing. Yes, so I see Tyler, a question about base editing. Yeah, so base editing is trying to correct the DNA. So yeah, absolutely. That's that's some work. Miriam Meisler is doing really, really nice work on that, on CRISPR, and trying to figure out like how much do we have to change to be able to have effect. So there's, but this is still very experimental in terms of doing this in mice, and trying to do this type of editing in the brain in patients is still like a ways away. There's lots of efforts to get this going for sure, and this is definitely something that's worthy to invest in research wise, absolutely. But I do think it's important to understand that would take a while to get to patients too. So that doesn't mean that it's not worth doing and trying and understanding, but it's still very much at the research level. I think with ASOs, and I'll get now to kind of the second way that ASOs can be used. I think ASOs definitely have a much quicker path to the clinic and to getting into patients, and because it is an accepted technology, and because you can just inject it into the spinal cord and it goes to the brain, which again is relatively easy, even though that comes with challenges. But So now we're going to move to another way in which ASOs can be used, which is what kind of my lab is working on, and also highlight some approaches that I think someone should work on, which if my lab can grow a bit, we can work on. But these two approaches to use ASOs in a different way. And so this use of ASOs is dependent on alternative splicing. And so I need to explain this biological process to explain how these ASOs can work. And so alternative splicing is this mechanism in cells that from the same piece of DNA allows different mRNAs and different proteins to be formed, depending on which parts of the gene get used. So each DNA region for a gene has two types of regions. The intronic regions, which are the black lines in between these boxes, which are exons. The exons are the ones that contribute to making the protein, and the introns actually just get cut out by the process of splicing. And so when DNA is turned into this initial pre-mRNA, sometimes the cell can pick which exons it wants to include. So you could have, from the same DNA and the same pre-mRNA, you can have an mRNA that contains the blue exon, but not the pink exon and not the purple one. So this contains these 4 exons versus another mRNA that might have the purple exon and the pink exon and the purple exon, but not the blue one, right? So you can have different versions from the same mRNA. This is called alternative splicing because the splicing, the removal of the exons and introns happens in alternative versions, which then leads to different proteins. So here you would have a protein that has the blue part, and here this one has the pink and the purple part. So this is a process that happens in many, many, many of our genes. This regulation of this is really important in terms of how our proteins function. This is actually what my husband's lab has been working on for 20 years. So this was kind of a great area for us to work on. So how does this apply to SCN8A? So this is a picture of the SCN8A channel, which weaves in and out of the cell membrane here many, many times. There are two regions within SCN8A, exon 5 and exon 18, that undergo alternative splicing, and that this alternative splicing changes the function. This happens in all sodium channels, so this is not just an 8A thing. And so I'm going to talk about both of these and how both of these can be taken advantage of therapeutically to regulate SCN8A levels and the level of the protein. Okay, so the first one we'll talk about is exon 18. And exon 18 is what we call a poison exon. And so in this gene, we have two versions of exon 18, 18N and 18A. This actually stands for neonatal and adult. If 18A is included instead of the N, so you have the green one, you get a functional protein. However, if 18N is included, this gets degraded. The mRNA gets degraded by a process called nonsense-mediated RNA decay, NMD, and you get no protein being made. Okay, so depending on which one is included, you either get your normal protein or you get no protein. So one therapeutic approach to try and take advantage of this could be to develop an ASO that would prevent the inclusion of 18A, of the green one, so that only 18N is included, and therefore everything that is made from this gets degraded, and this would reduce the levels that we would want. So this is what we would want for gain-of-function mutations, right, where we're trying to reduce the levels. So if we could develop an ASO that increases the inclusion of 18N so that you have a lot more of all this mRNA gets reduced and degraded. So it's a slightly indirect way of doing this compared to the ASO that directly degrades the mRNA. And there's a lot of belief in the field that this could be a gentler way of reducing SCN8A, right? That the other approach is really brute force, degrade everything, versus this approach is switching and inducing degradation in a way that is more gentle and might be more controllable. So it might be less efficient, but that's a good thing because we don't want ASOs for SCN8A to be too good because then it's too hard to control. So we want ASOs that are just not that great, that work well, but not that well. And this could be an approach. And so there was a company that was working on this and they shut down for complicated reasons. But no one is working on this right now. And I think this is something that we should be doing in our community. The other flip side to this is that this could actually be used for loss of function mutations too in a different way. So here, if we developed an ASO that instead prevented the inclusion of 18N and included 18A, all the mRNA would be turned into protein, and this could increase the levels that are present. Okay, so instead of having some degradation, we can tune it up a bit. And that's what we want for our loss of function patients, is make more. And this is the approach that the Stoke Therapeutics is using for SCN1A. You may have heard that there's a trial ongoing for SCN1A, which again is largely a loss of function disorder. These diagrams basically show exactly what I just showed you. And again, because SCN1A also has this exon 18. And so what they're have developed is an ASO that prevents the degradation of SCN8A so that it brings the levels up back to a normal level. And it's in clinical trials. They've released some data that they're seeing some effects, especially in children that they inject that are younger. And not just some effects on seizures, but also some effects on development, right? Because here you're not just like blocking the activity of channels, but you're trying to correct like the level back to how it's supposed to be. And so I think these kind of treatments are also pretty exciting because it's not just dealing with the symptoms, it's hopefully actually like dealing with the underlying cause and maybe could have more effects on development, behavior, these things, that, are obviously also all equally important to us and to our children that, with just blocking seizures or kind of just addressing the symptoms don't really get at. So we still don't know, right, exactly how much of an impact these therapeutics can have in the brain. That is a huge unknown. How late can we give these to someone to have an effect? All these questions are things we don't know, that we are learning. And these trials, this Stoke trial is really exciting in terms of that. And so, but this approach is very much doable for SCN8A, especially for loss of function, and I think maybe also for gain of function. And so, again, I think this is something that, we really need to be working on more. And again, there was a company, but unfortunately for complicated reasons, it did not, it is not kind of alive anymore. And so, I think this has a lot more potential, again, because this might be like slightly less efficient and we could control it better and have less risk of side effects.
I was just going to jump in real quick. And so with that, would there have to be two separate ones, one for loss of function and one for gain of function, right?
Yes.
Yeah. But they wouldn't be based
Absolutely.
on each mutation. It would just be gain of function or loss of function.
Exactly. Yeah. 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 So yeah, like the one from the Stoke one, they were able to make an ASO specific that is not dependent on the mutation in any way. So yeah.
Cool. Thank you.
Any other questions on this? Hopefully that, I know it's a complex biological mechanism, but I think, this is really exciting. And because this is happening for SCN8A, so this is something that, we should be able to make safe ASOs that do this for SCN8A. We just need to test them really rigorously. If it passes the trial, can it be applied to loss of function SCN8A? The SCN1A ASO, no, I don't think so, because no, not the one that's approved. That one would not work for SCN8A patients. We have to make our own one for SCN8A. Yeah. Okay. And the last approach that I'll mention is it's a similar approach, but with this other exon 5 region. So again, this exon 5 region, there are two versions, 5N and 5A. And actually, over time in the brain, this is the levels of each of these in the human brain and in brains before birth and after birth. And so before birth, you have a lot more 5N, and then it switches over time to become 5A. Now this happens, again, in all sodium channels. No one really knows why or how or what. So this is some of the research that we are doing. But we know that this switch impacts the function of these channels. And so this is a recently published paper from Al George's lab, and he has a similar study coming out for SCN8A as a teaser, which hopefully he'll present. But what he showed is that they looked at whether having either 5A or 5N in the gene impacts how a certain mutation affects the sodium channel activity. So, for example, this is the R853Q mutation in SCN2A, which in 8A is the R850 mutation. And you can see these are features of the sodium channel, so how open they are. And so you can see that when there's the adult version, the A, in NAV 1.2, this is SCN8A. When the mutation is present, there's a difference between them. But when if the neonatal exon 5 is present, actually that mutation has no effect on this parameter of how open the sodium channel is. So again, when there's the adult, the mutation has an effect because the lines are different. When the neonatal is present, there's no difference. So there is some evidence that this switch that happens, in one part of the protein could affect a mutation that is happening in another part. We don't fully understand, again, how this works, but it could be that switching between these two exons, could affect how severe a mutation is. The other thing is that there are many patients who have mutations in this region, including Margot. And so if your mutation is in between 206 to 236, you are within exon 5. And that means your mutation is either in the adult version or in the neonatal version. And to know that, you have to look back at your original report. It doesn't say it because this is not something that a lot of geneticists look at, but we can find it out. And if you are in this region and want to figure out, and I'm happy to talk to you about that. But each purple arrow is a residue in the patient registry that has been shown to cause seizures, to cause a phenotype. And for some arrows, there are multiple patients. So there are, I think, about now 40 to 50 patients that have mutations in this region, and one of them is Margot. So our approach for these patients, and this is what we're really actively doing in the lab, is that if a patient has a mutation in the exon 5 in the blue box, then we develop an ASO that prevents exon 5 from being present in the protein. And instead, we replace it with the correct version of exon 5A. And so we have a correct protein where the mutation is gone. Right? So we're not touching the DNA, but just by switching the RNA, we can correct it. Similarly, if a patient has a mutation in exon 5A, we can develop ASOs that switch it to make it 5N to correct it. So again, for patients that have mutations in this region, we are hoping and we are developing tools to be able to correct this completely. And some data from the Al George's lab suggests that changing this could also impact other mutations in other regions. So it could be that inducing a switch in this way could have effect on mutations in other part of the gene. We will need to test that in mice and things. But right now, at least for these patients in this region, we could potentially correct it. And so I'm going to show you some data that we've collected in the lab that we can do this. So, initially we thought, could we even do this? But we can. And so this is all work by a grad student in my lab, Haley Dame, who I recruited to work on this project with me. She's working on this. So we use different cell lines to be able to measure the ratio of each of these exons. So 5N in blue, 5A in red. And so different cell lines have a different baseline level. And there's a question on, is there a large difference in animal models that are 100% 5N, 100% 5A with wild type SCN8A? Yeah, this has not really been looked at in any way. So we are hoping that these ASOs we've developed can allow us to look at that and ask that more precisely. But we don't know. That hasn't been looked at properly. So then we wanted to test ASOs to see, okay, can they switch from one to the other? And so here I'm showing you actually some gels, some DNA gels. So we don't need to discuss the method, but I'll just show you what this looks like. So if you run our control cells that are not treated, the box at the top is 5A and the box at the bottom is 5N. And so the more abundant this black band, the darker the black, the more of the 5N there is compared to 5A, which is very faint band. And so we tested ASOs that we designed. And so these ASOs, you can see, have no effect, right? They look exactly like the control. The band is really thick at the bottom. The band at the top is really light. There's no change. But then we found some that worked, that induced this flip in the switch. So Here we removed completely this band at the bottom, and instead we have this huge band at the top. And so we're able to remove all the 5N and switch it all to 5A. And so we tested 18 of these, and here's the whole data. So here's the control, where in the control cells, we have mostly 5N in these cells, but then you can see with certain ASOs, they switch in the other direction. So this is really exciting that we have tools that do this, and we've tested these in lots of different cells to make sure that they work. We're also looking in the other direction. And so we developed ASOs, and this blot is in a different cell line where we have, this cell line has about 50-50, a little bit more 5A than 5N. 5A is at the bottom here. You can see in the control, you have, but the same intensity of band in the red box versus the blue box. But when you treat with the ASO, the band is completely gone in the red box, and it shifts up to the 5N. And so this is shown in the graph here. So we have tools that can do this, that can switch from one to the other. And so we're working on using those. Wait, sorry, this is where I want to go. So, what we are doing is we are testing these ASOs in cell lines that have Margot's mutation. And we've also generated a mouse model that has Margot's mutations, because there are no mouse models of SCN8A that have mutations in this region in exon 5 or in this region of the gene. All the mouse models that exist so far for SCN8A are in other parts. Thanks to funding that we got from the American Epilepsy Society in partnership with the Acute Syndrome Foundation, we are characterizing these mice with our collaborator, and we're hoping to test these ASOs in this model. Try to develop this approach for both switching in one direction and in the other direction. But so overall, I think ASOs have a lot of potential for SCN8A. Mostly in terms of the splice switching ones that I talked about for specific mutations, or with this poison exon area. I think that is something that would be really exciting to pursue and see if the approach kind of, this approach that kind of Stoke has taken with SCN1A, if that's something that can be done. And if that could be a better approach than, this these direct degrading ones, which no company wants to touch, unfortunately. And so with that, I'll just acknowledge again Haley, who did all this work in my lab and all the funding that we have. And this is my husband, and this is us at the gathering last year, with also Chris Dula, who's our Tufts epilepsy collaborator, who popped in for a little bit when he was at AS to say hi. So this is the team that is working on this right now. And yeah, I'm happy to take more questions, and hopefully this kind of cleared, kind of explain a few things about ASOs and how they work, and where we're at, and what can be done and what cannot be done, or what is more challenging to do. And yeah, I'm happy to, answer questions now or later. You can find me on Facebook or Instagram or in person at the gathering.
Exactly. Thank you so much. I feel like even I could listen to this probably 5 or 6 times and learn something new every single time. Or, have something that clicks that maybe didn't click the first time I listened to it. So thank you so much. Can you, okay, so you're working on this with your lab, and I think everyone's like, oh, this sounds really exciting. Like, can you maybe give us a short synopsis of like, what has to happen from where we are now to actually like getting it into humans or our kids?
Yeah, So, I mean, there's a lot to do, because the main thing, is that academic labs, like, we can do lots of research to show that this can work. We can do the switch. For example, we can test this in a mouse model. We can hopefully see that it might improve seizures, for this, that this is something that can work. But then, I don't have the capability in any way to take this into patients, right? I'm not trained to do that. You don't want me to do that. Like, I'm an academic researcher. And so that's where then, we do need pharma companies to be doing this, because usually what pharma companies would do is like, we, screened like I said, when we were looking for these ASOs, we usually what you do is you screen a lot. And so we screened 18 because we're an academic lab and these are expensive and we don't have the resources to do this. But in a company, when they're trying to do this, they screen 500, and then they look, okay, and then they'll identify the ones that work best. And then the main thing with ASOs is that they can be toxic, right? So because of the nature of the biology of what they are, so then, there would need to be toxicity studies. If you find good candidates, you would usually test 20 or so in mice and rats and that, again, needs to be done by people who are qualified to do this. And then, technically then you can go, if you have a safe ASO that works, then you can go to the FDA. Now, with Margot, is a patient in the NLORM Foundation. So we are working with this company, the NLORM Foundation, which is spun out of Ionis, to get a switching treatment working for Margot. So, and tested in her. So this is kind of, again, initially on an N of 1 basis, so for Margot, but she will be our guinea pig to see can this work in patients with SCN8A, right? We don't know. And how much of an effect can we have, right? Because I think especially with our, really severe kiddos who, have very limited skills, it's at what point can you switch something in the brain to see an effect, right? And that's just a question we just don't know, and we still have so much to learn on what can we change in the brain at what point in time to see effects. So we are hopeful that with NLORM, Margot will be injected within the next year or two. And obviously we'll learn a lot from that in terms of what can be done. But, to then develop this to more patients, we would need a biotech company to take this on and to kind of develop this more towards clinical use in terms of toxicity and safety and all these things, and do kind of a more rigorous, in-depth, all those steps that are needed to get this into patients. But certainly, academic labs all can, often do kind of these experiments to show that this can work, right? We can do this. When we showed, and NLORM that we could switch this, that got them so much more excited because they're okay, yeah, if you can do this, then, this should be able to help. And so I think, and so, in terms of these other ASOs for the poison exon part, there is a patent, which is public, so can share about it. It's not proprietary, where they have some ASOs that do work and that change this poison exon. So there's not much else that's public on this. So there's lots more experiments that needed to be done. And some of that can definitely be done in an academic lab initially as, again, more of a proof of concept to show, can this work? How safe is this in kind of a basic model? But then, we do need pharma and companies to take this on, to take it to patients, right? I can't do that. Academics can't do that. And you don't want us to do that because we're not trained to do that.
Yeah. I was going to look and see if there was any other questions. Essentially it was a lot of people just saying thank you and that, being able to break it down. Tammy has a good question. As a community, what can we do to help? where in this process?
Yeah, I mean, I do think, there's some good work to do on on doing more on these ASOs, kind of the Stoke ones. So either even I don't know where Stoke is at in terms of taking other genes or something, but I think if there could be some experiments in a lab to test, kind of show more proof that this can be done. This can be done in certain mouse models. There is a loss of function mouse model out there. And so, I think investing in that in research wise would be great to be able to, get more data on that, at least in a lab, to be is this convincing? Is this, can we do this? Is it worth for a pharma company to come in and really take this on, So I do think that's exciting to look at and to do as well. And I still think investing also in like I said, the research that the Meisler lab is doing on CRISPR and stuff, there's so much to learn from that. Again, it's therapeutically, it's a little further away. But we have to invest in all that basic research to ultimately lead to treatments. So how do we try to connect with the researchers? I mean, I know some of them and many of them will be presenting and attending at the gathering and stuff. And so, those are good ways to connect them and email them. And I know Miriam will be presenting in a bit as well. And so maybe I'll message her and be like, people were asking about this, you should talk about that. And be like, you should present on this. She's done such great work in pioneering a lot of this. And so, so she's also a good person, but yeah.
Awesome. And then just since this is a little bit of a teaser, so you will be chatting with us again, and we don't have it won't be live streamed, that family education portion, but I think we can easily get something set up to where we can record it and share it later. And so, what are your plans on as far as building on this when we chat with you in Orlando?
Yeah, I think I'll definitely kind of reiterate a lot of these concepts and expand a bit more on some of the stuff that we're doing in the lab and kind of how we get things going. I think, as you said, it's always good to hear these multiple times. These are complex concepts too, even. And so I do think it's good to then, be, and I hope also to kind of give an overview, where all the different drugs are at so that all the families can be primed for the evening and ready to ask questions to people. And maybe I'll put up some pictures of if you want to ask this, go talk to this person.
Right?
And so the goal is also to kind of make sure that you're prepared for the evening session and can make the most out of that in terms of who to go talk to and ask questions about. So it's also, kind of like a preparation for what will then be presented, in the evening session, I think.
Perfect. Yeah. Awesome. Well, thank you so much for doing this. And we will see you in Orlando.
Yeah. So soon.
Yes. So thanks everyone that joined today. And thanks again to Neurocrine Biosciences for supporting us in this series. And then next week, we will have 2 researchers out of a lab at the Children's Hospital of Philadelphia. And then the following Wednesday, then we'll have Miriam Meisler on too. So super excited to kind of have more of these one-on-one conversations where we feel super confident and comfortable asking questions. I know sometimes in the gathering, it can get a little overwhelming to raise your hand in front of a room with, 100+ people in it. So I'm really excited to be able to have these kind of smaller conversations where families can really feel comfortable asking questions. All right. Well, thank you again, and we will see you soon. All right. Bye, guys. Thank you.